Hello, my name is Cindy, and I will be discussing the topic of defective CFTR. What is cystic fibrosis? It is an autosomal genetic disorder that is caused by a mutation in the gene that codes cystic fibrosis transmembrane conductance regulator proteins, or CFTR. CFTR encodes for CAMP regulated chloride channels that are located in the apical membrane of epithelial cells, especially in the lungs. It regulates the passage of ions through the membrane. Cystic fibrosis causes increased viscosity in mucus. It alters the ion transport channel and can lead to severe bacterial infections. Some of the main organs that are affected by this disease are, of course, the lungs, sweat glands, salivary glands, pancreas, liver, or really anything that secretes anything. There are 7 different classes of CFTR dysfunctions. I went ahead and listed them here for you to see. I've also listed what each defect does, or which class. In this slide, the image is taken from the article Cystic Fibrosis, Current Concepts, which is published in 2021, and it gives you more of an idea of what is going on in the cell membrane that is causing the defects of cystic fibrosis. You can kind of see here and read what each does. There are many impacts that defective CFTR has on the individual with cystic fibrosis. Some examples include, as mentioned before, the thickening of mucus in the epithelial lining of the lungs. Defective CFTR cannot be processed by the endoplasmic reticulum. There is an improper balance of salt in the epithelial cell linings of the lungs. No normal chloride ion transport across the membrane, as there is in healthy individuals. There is an increase of chloride ions and water molecules inside the epithelial cells, and there's no hydration of extracellular mucus and secretions. I've included an image here to give you more of an idea of how a mutation, such as the one seen here, which is a class 2 mutation, where the protein is made, but due to the misfoldings, the protein cannot reach the cell surface. You can easily see how there is an accumulation of sodium and water in the cells, and how chloride and bicarbonate cannot leave the cell. There is also that increase of the viscous mucus on the outside of the cell. In the last slide, I describe how gene therapy is able to help fight cystic fibrosis. CRISPR technology is being used to edit the genome of patients with cystic fibrosis. It is used to help repair CFTR locuses. Here on the right is an image taken from gene therapy of cystic fibrosis. It lists some of the techniques used. I'll go ahead and break down the colors for you. Orange indicates the CFTR gene that has a mutation. Blue is going to be the correct sequence. The elongated bars indicate 27 exons distributed over the entire length of the CFTR gene. And the arrows here that are pointing to the right indicate the transcription process. In the center of the image, which is all orange, this represents the patient with cystic fibrosis. I've gone ahead and listed how each of these steps work in some detail, but I honestly recommend reading the article. It was very interesting because it kind of gives a more in-depth of how these techniques are being used. This last slide is just my reference list. Thank you so much for listening.